Hey Biology 11, Mr. Bleeker here. Can you believe it? We're at Chapter 22, Section 5, which is all about the angiosperms. And it's taken us a bit of time to get here. Plants had to invade land, and we looked at the bryophytes, and they didn't even have water conducting tissues. We moved on into the ferns, the pterophytes, and saw the rise of the water conductive tissue, tracheids, that's a class of xylem. And in our last lecture, we looked at the gymnosperms, the gymno, naked, sperma seeds, the ones that don't produce flowers and they don't really have fruit. Now, ginkgo biloba was kind of interesting because there was a fleshy seed developing there. Now, what we're going to do is we're going to get going with these ones, which are literally entitled angio, which means in a vessel, sperms, seed in a vessel. You get how that works. And there's an incredible interaction with the angiosperms and the rest of the world. In fact, they're highly successful because with the deliverance of flowers, they brought in a whole bunch of cooperation, not just flowers, but fruit. They brought in mammals, birds, insects to help them to disperse their seeds. The next time you eat a banana, there's little seeds in there. Think about where that goes through your digestive tract. You're dispersing seeds. When you watch a black bear in the forest consume berries or whatever it can, it leaves behind the seeds in a little pile. The fruit is really a driver, it's a mechanism for animals to disperse the seeds of the plants. In other words, it's a mutual relationship going on there. Mutualism. And that's a good thing. The evolution of our color vision is arguably even in tune with flowers and fruit because we have little cones at the back of our eyes. They're in our retina, and they can differentiate between red, green, and blue colors. And our ancestors developed the ability to tell ripe fruit by developing color sensitive vision. That's how important plants are to us. They keep us well fed, and they're some of the most nutritious things on the planet. God bless those apples, oranges, and everything else. Okay, so we're going to get going here. When you go to the site, you'll see that there's a huge amount of resources here. In fact, I've got multiple lessons that go even further than the book or the book does, rather. There's a bounty of things I want to show you. And it's nice to have a digital course because I can get into that and you can take it in your own time. So, there are wonderful videos here. You need to go through these. And a whole array of animations. And this will all make sense because by the time I'm done, I will have gone through a good number of them and explained them. So let's get going. I've got reflection on, and like I said, let's get this started. There we go. Flowering plants. Now, with flowers came fruit. The idea, and if you ever look at a tomato plant, for example, the idea is to, let's move that down a little bit, and let's choose a little bit of a green color. Maybe not that light green. Let's go with that. There we go. Look at the base of a rose or at the base of a tomato plant, just underneath the flower, and what you'll see is sort of a green area down here. And there'll be a nice floral portion on top. Let's have to hit done, switch back, get a nice color for this. This won't be my best flower, but it'll be a quick one. And the flower itself, pick a nice color. Let's go with some yellow petals. The flower itself provides these petals, these enticements to animals and insects and things like that. Because what it wants to do is down here, the eggs are contained in ovules, and it wants to fertilize those. So in order to get them fertilized, the flowering plants are inviting in animals, and of course, wind can help with pollination. But now, when we look at some of the insects, and it can be highly specific, bees and moths, etc., they deliver the pollen in a way that the pollen is able to get at the ovaries. So introducing one gametophyte to another. The pollen grain meets the ovule, which contains the egg, and we get fertilization. And here's a hint. Wherever you see a flower, you'll see an ovary somewhere adjacent to it, just underneath. It's considered to be a feminine structure. There we go. So we'll draw a little female sign there. And the fruit, what that is, fruit is when a seed with its endosperm develops, but is inside, usually, a swollen ovary or ovule. I'll just say ovary for simplicity's sake. And this, for example, when you eat an orange, is the piece of the orange that you consume, and then there's a seed that you toss out. It's all an enticement, folks. It's a smart one. So the flower is a reproductive organ. Absolutely. As we said, it was put there to attract animals. And by attracting animals, largely what that's about is pollen transport and to get it from flower to flower. That's a smart strategy. There's a couple videos on the course website you'll need to see. Some of these relationships between some of these flowers, if you think about the yucca plant, for example, the yucca plant has a relationship with a bat, which is amazing. When you look at some flowers, they only have relationships with certain species of bees, and very particular. Interesting how highly specific it can get. But I got a few videos that go over that, so that's pretty neat stuff. The flowers themselves, they contain ovaries, and inside of the ovaries are the seeds, which we just talked about. And the pollination is an interesting process because you would say, wait a minute, I thought the pollen fertilized the egg and produced the seed. Now, it's a little bit beyond the scope of this course, but there's something called double fertilization, where not only does the seed get fertilized, but the there's an additional structure outside the seed, which also gets fertilized and develops the fruit. So there's actually two fertilizations going on, one for the critter and one for the structure that develops into the fruit. We won't go that far. It's called double fertilization. It's very neat, but like I say, it's just a little bit beyond the scope of the course right now. But if you're interested, check it out, double fertilization. Now, in order to understand the angiosperms, you should know about flower anatomy. Okay, this is kind of interesting, but I think I'd rather animate or annotate something a little bit different. So I'm going to pause this for just 1 second, show you a neat little opportunity that I have for you here. And it is right here. Okay, I'll zoom in a little bit. There we are. So if we're looking at the structure of a flower, what you have to understand first about petals, I'll talk about them. Petals, and if you look at a rose, for example, it would have the greenery underneath, these things called the sepals. Sometimes that greenery on the underside can be on the underside of a petal. Sometimes it can be separate. But the point is, petals are just modified leaves, and they're very attractive, and they're designed to draw in insects, bats, mammals, etc. They can be quite fragrant. That's part of it. So the petals are modified leaves. The sepals, these things down here, are protective, sometimes fused with the bottom of the petal, sometimes not. The male structure up here is called the stamen. And the easy way to remember that, I always tell my students, think about sort of masculinity, strong, having stamina for sporting activities. The stamen is the male reproductive structure, and it's smart of this plant to hold it above this structure here, because what you want to do is you want to make sure either hold it above and ripen it at a different time, or another strategy is for it to be shorter so that you can't accidentally self-pollinate. We saw this kind of issue come up when we were looking at the fern. You don't want your reproductive structures to develop at the same time, because if the pollen does get released and it lands on the female structure, which is right here leading down to the ovary, lots of little ovules in it, that would be self-fertilization. That would not be a good idea. So the idea is to get insects, wind, or some vector of transportation to come into contact with these structures here. They're called anthers. Let's see if we can zoom in a little bit more. There we go. With the anthers. And this is undergoing meiosis and producing your pollen. So your pollen grains in there, your gametophytes. So this is in a male, let's use an analogy. We'll talk about humans. It would be the testes where this would be going on. So the anthers, that's the actual structure where the pollen is being created. The filament, it just holds up the anthers. It's actually the anther that's the reproductive structure. Okay, so that's the male part. It's called the stamen, and it's made up of the anther and the filament. Let's see here. The pistil, I always think about a female gunfighter when I think about this, right? And if you cheat on them, they'll shoot you, right? No, I'm kidding. But it's a neat analogy, so I'll just run with it. Got to have a little fun in this job. The pistil right here is composed of the sticky stigma. So if you ever run your fingertip over top of the stigma, it's a little bit wet, a little bit sticky, and that's meant to trap the pollen grains. And their job is to grow down through pollen tubes, down, 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 until they can get to the eggs down here in the ovary and fertilize not just the ovary but surrounding tissue, and that would develop fruit. Tomato plants, anytime you get fertilization down there, you get the seeds being developed and the ovary itself swells up and you get this amazing production of fruit. Okay, so you got your sticky stigma here, then you've got your style. Okay, style, right? It's basically this neck-like structure that leads down to the ovary. And the whole thing is called a pistil. Stigma, style, ovary. Good. Pretty well got it. And we'll look at all the structures. There we go. And we can just sort of do a little self-test. Petals, sepals, ovary, style, stigma. Okay, so there's the pistil and anther, filament, male structures. Pretty straightforward. Not a bad little animation to take a peek at. Now, interestingly enough, I'm glad this came up next. When you look at insects, they've adapted their body structures to provide an opportunity for themselves over time to have an interaction with angiosperms. Now, this, it's funny how they give it this name, but when you look at this, right, we're looking at a moth here. A butterfly would be very similar. This mouth structure right here is developed to lower down, to uncoil, to go down and get the flower's nectar, right? That's what honey's made from. Honey, in case you didn't know, is highly concentrated flower nectar. That's why there's many different types of honey. It's a sucrose solution, but it's a little bit different from flower to flower. So we tend to love all the varieties of honey out there. And the mouth structures are adapted to get at that. You can see the highly specific relationship between a butterfly, a moth, and the flower in general. Now, another thing, if I always think of if you've ever seen the bee movie, they will also, some of these critters will actually feed off some of the pollen grains themselves. But the nectar is extremely nutrient rich. It draws in insects. When you look at mammals and birds, they're highly attracted to the fruit per se, but there's many ways that plants get us interested in them. In fact, if you look at this little doodad I have for you here, humans can only see in what we call the visual spectrum: red, orange, yellow, green, blue, indigo, and violet. Now, what do you think insects see? It's a little bit of a presumption to think that they only see Roy G. Biv. In fact, they see colors we don't. You might have heard of colors that go past violet. There's a spectrum called, well, not a spectrum, but there's wavelength. There's a wavelength called ultraviolet. Check this out. When viewed with a special camera, all of a sudden you see very interesting patterns take shape onto flowers. A lot of them indicating, hey, more or less, come check out the middle of this structure. This is where the reproductive part of the plant is, and it's sort of welcoming with these patterns, or recognizing that this is where the insect or the well, we are going with insects here. This is where the insects should show interest. And you see this again and again in what these patterns look like with a special camera, special visual aid. It's just fascinating to see what insects view and what we view. This is Roy G. Biv, and this is a very different view. So it's very inviting, and for a reason. The plant is trying to reproduce, and it's trying to partner with the insect in a way that makes that ideally successful. Very cool. Okay, so I'm going to pop back to the main portion of what I was going over. Thanks for bearing with me. And there we go. Now when you look at this structure, you can say, okay, I've got it. Little extra labeling wouldn't hurt. Stigma style ovary. We'll just call that the pistil. Female gunfighter part of the plant there. And the anthers in the filament right there. Let's make sure we put that label in there. There we go. And ask yourself a little question. What the heck do you call that again? Oh yeah, that's stamen. There we go. We've got the petals. This time they've not just called it an ovary, but they're looking at the specific egg chambers inside, right? The ovule. You don't hear it called archegonia anymore. Okay, those are the ancient egg chambers. Really, about the time you move past the ferns, you don't hear that a lot. Okay, the receptacle, in case you're wondering, it's just a little bit of extra information here. That's the base of the ovary. And the pedicel right here, I'm okay if you think of that as the stalk. That's not a big deal. There's a lot of diversity in the angiosperm kingdom. If you think about biology, and I always remind students, you have to be a biological librarian. How are we going to sort out this enormous group of angiosperms? Like, it was hard enough with the gymnosperms. We had the ginkgophytes and the gnetophytes, and it went on and on. Now with the angiosperms, holy smokes, well, how do we break that down? There's so many types of flowering plants. Well, it turns out you can pay attention to the patterns that are presented to you. From the time they grow up, how many leaves do they stick up? Are they woody or are they kind of soft? Do they have are they herbaceous plants? Do they live 1 year? Do they live? Does it take 2 years to complete their life cycle? Are they long-lived? But an angiosperm is kind of interesting because there's a lot of categories they can belong to. We're going to illuminate this now. The first way you want to sort them out is monocots and dicots. Now, by cot, they mean the cotyledon. So I'm going to zoom in a little bit here. When this little baby plant is born, it's born with embryonic leaves. So more or less inside, it's a little sort of, let's see if I can get my, there we go. It's tucked away inside, and it's got a little primitive root down there, undeveloped root. And once it begins to grow and germinate, the roots are what we say, they show geotropism. Tropism means preference, and geo means earth. So some of these roots will grow right down towards gravity. And that's good, because they need to seek out nutrients in the soil, water, etc. Now the stem itself, and the leaves for that matter, are phototropic. So this is a geotropism. A little information here. And let's zoom out a little bit. There we go. And our primitive leaves are cotyledons. We'll just use the singular, cotyledon, is phototropic and aims up towards the sun. Now, if you don't believe that, you can flip this little guy upside down, partway through an experiment. Just turn him once the roots begin to develop and go downwards, and you'll see that the roots will curl right back around and they'll try to grow down. But you should prove that for yourself. Prove that the geotropism exists. Phototropism, once a stem starts to grow, you could turn this whole structure upside down. Might be a little hard. In some loose soil, you could do it. And see if you turn it upside down and inverted it, if it would right itself in its growth pattern. It's pretty neat. That's a real big deal when you consider growing plants in space. If there's no gravity, what's the primitive root, the radical, supposed to do? That's why they've been doing experiments up on the space shuttle to see what it's like to grow plants in a lower zero gravity environment. But I digress. That's normal for me. The cotyledons are very primitive. They get a little bit of photosynthesis going on. The plant has to hurry up and start feeding itself, and then the foliage leaves kick in. And those are kind of like generation 1 leaves. And what we have is our developing embryo. So over here, you can see it developing and doing what it needs to do. Now we're talking about monocots and dicots. So I was stressing the importance of the cotyledon, the embryonic leaf, or leaves. It turns out that's a really neat way to differentiate flowering plants. Is it born with one embryonic leaf? Is it born with two? That's why we say we've got monocots. Let's bore this. Monocots, which have one embryonic leaf. Or are we looking at dicots, which have two? Now, an easy way to remember this, and I'm going to do this right now, is I always remember that corn's got mono. I know it sounds ridiculous. Mononucleosis, that if you've been a teenager, I've known a great number of teenagers that have it, where it makes you feel really tired. It's a viral infection. Okay, think of it this way. Corn's got mono. It's so ridiculous you'll never forget it. What this means is that corn is born with one embryonic leaf. So that's a stock example of a monocot. Now, if we grow up a dicot, a stock example of that would be a bean. And there we go. Monocots and dicots, two of the simplest examples. I tend to get that out of the way right away. I don't want you to get confused. Okay, so what are the characteristics of monocots and dicots? Well, if we zoom in on their seeds, monocots are born with one cotyledon. Corn will come up with just one little leaf, whereas bean will come up with two. And you have to look really close at the diagram. This is trying to indicate to you that there we go, that there's two little baby cotyledons there. Whereas in corn over here, it just has the one. I think they're trying to indicate it being right here. Just one little simple cotyledon. Now, if you look at the leaves, that's another dead giveaway. In something like corn, or grass for that matter, what you'll see is that there's these striations that go down the leaf, and they're all in parallel. So I'll just do a little markup here so you can see what I mean. They run parallel to each other. Grass is like this. Corn is like this. And that's another giveaway that they're kind of in a similar group. They have a similar characteristic. And as soon as I see this, if I see parallel venation, I'm pretty darn certain that this little critter only had one embryonic leaf. Kind of all goes together. If I see that the leaf has what we call a branch structure, kind of like think of it, the veins in the back of your hand. They're netted, and they go like this. Netted venation. A little bit more random like that. Well, then you can say, oh, that's a dicot pattern, and this plant would have two embryonic leaves, similar to a bean. Another thing you can do is look at the floral patterns. Monocots are characteristic for loving this multiple of three. Okay, 1, 2, 3, 4, 5, 6. Their floral patterns tend to be in multiples of three, whereas dicots will have floral patterns in 4s or 5s. So in the lab, in my classroom, I was going grab an African violet, for example, and it's such a telltale sign. I have a student pick it up, and I say, hey, count the number of you have to do it a couple of times to make sure that you it didn't just accidentally lose a petal, but I say, go ahead and count that. How many leaves does that have? Is that a multiple of 4 or 5? And then I can say, you know what? African violet, it's got branched veins. Big surprise. And then I say, well, how many embryonic leaves do you think that thing had? Well, scroll so I can actually write. There you go. It probably had 2 cotyledons. So these characteristics tend to give you some really good organizational abilities. I'm going to pop out of reflection. There's a little bit more to it than that. I think it was a little oversimplified in the presentation. So there's a little bit more that I wanted to show you. And I'm just going to mute this because this drives me crazy. But now they call this a eudicot. EU means true. It's a little bit more than you need to know. This is a dicot. There's the 2 embryonic leaves. And here's corn with the 1 embryonic leaf, the cotyledon. Okay, floral patterns, right? There's our monocot flower, right? Pattern of 6. Over here, like I said, like an African violet, patterns of 4 or 5. There's your monocot leaf with the parallel veins, and then the netted hibbledy-gibbledy pattern over here with the dicot leaf. There's more. The pollen grains here will only have one of these little openings, whereas dicots will have patterns of multiple. Now, I'm going to have to pause this. When you look, if we think back a moment ago when we were looking at the corn leaf or grass leaf, the reason that it has all those veins running side by side is because if you cut through the leaf or if you cut through the stem, you'll see that these little bundles, they're called vascular bundles, they conduct the water and the sugar in the plant. That's exactly what those little striations, those little veins are. They're pipes. And I'll get into this in more detail when I talk about how plants transport water and how they store their sugar. But think of these as veins, okay, as plumbing. And there's 2 types of plumbing. There's food plumbing, okay, which is food phloem tubes. And there's xylem, which is water. Now, nobody said those pipes were going to be, separated from each other by a long distance. In fact, they grow very close together. And if you look at the veins or that pattern in a corn leaf, the reason why they're all they never touch is because they're evenly distributed and scattered. But you can see that they're all in parallel, kind of like railroad tracks, right? They never touch. Now, that is not the case. That is not the case when you look at a dicot, right? Dicots don't set up their plumbing to be like this. See how it's distributed all throughout the stem? These are very interesting. They set up their plumbing, their major plumbing systems, around the stem in a circle like this. So scattered venation, okay, all the way throughout. This is venation, okay, vascular tissues arranged in around a cylinder. Okay, give that a second there. I think I've kind of reached the end of it. Here you go. So this is just a little game to see. I'll put this on the site. You tell me if you can figure out if it's a monocot or a dicot, right? Can you figure that out? Yep, that's a eudicot. Count this. What have we got here? 1, 2, 3, 4, 5. Test yourself out. See how you do. I'm going to close this now. There's another thing that didn't get mentioned in your book, and it wasn't mentioned in that animation either. When you look at a grass plant, if you've ever had to weed the garden and pull up grass, and you pull it up, you notice it has an extremely fibrous root, and it's very frustrating, right? There's the parallel veins, 1 embryonic leaf. Oh, okay, I get this. But when you look down at the roots, they're fibrous and spread out, and they don't tend to go that deep, but that's a lot different than if you look at a bean plant where it sets up a principal taproot. Now, for those of you who know about dandelions, you go to pull them out of they have a different reason why they're hard to get out of the ground. They've got a strong central taproot, and then they've got the fibers going off. You don't see that taproot structure in something like a monocot. So that central taproot is totally dicot. Okay? All right. Enough said. In case you were wondering, by the way, on the course site what this is, if you've ever gone out into a field of lily pads, It's fascinating. They're an incredible looking flower. I always find it neat to kayak through those things. I wouldn't want to fall off though. Yee, weeds give me the creeps. Okay, so here we go. So the next half of your table has been pretty good about this. Okay, monocots with the veins, the plumbing scattered throughout like this. And it's one of the reasons, by the way, they can't develop woody structures because they're not organized. They've got their tissue, even though it's parallel, all hibbledy-gibbledy throughout the stem. But this is a little different. When you look at this, I'll give away a little bit of a hint here, and it's a little bit evident on here. You'll notice a ring here. If I draw it in, and that will come up in a later lecture. Oop, I lost my ring. Okay, that's all right. I'll draw it back in. It's right here. The reason that some plants can lay down woody tissue is that they tend to grow their xylem on the inside. They those specific pipes grow to the inside and get pushed to the middle. And remember, xylem is heavily reinforced with cellulose. Well, what's wood? If you think about a woody log and you see all those rings, that is xylem. And as it gets pushed in towards the center, the pith in the middle of the structure, it just gets crushed under the pressure of all the growth. The phloem, I'm just going to get rid of that P there. The phloem is this. And it tends to grow out towards the outside of the plant. And it's interesting because in different plants, depending on how long they live, etc., you can get xylem laid down each year, and then you get secondary xylem. And if plants are long-lived, you'll get secondary phloem. And that special ring that I had right there, it's called vascular cambium. What does that mean? I'll label it. Vascular cambium is capable of becoming cells that are sort of undifferentiated. They can become phloem cells or they could become xylem cells. Year after year, if it's a long-lived plant like a perennial, and if it's a woody variety, if it lays down secondary xylem again and again and again, those water pipes get crushed and they don't work after a while. But the younger ones, right, the younger ones that are kind of new, yeah, that's functional tissue. And it's because they arrange themselves in a nice ring like this, always growing the xylem in towards the middle, that we get that orderly production of xylem, and that's why it's the dicots that produce the woody tissue. So there you go. Now you know. Looks pretty good when you zoom out on it, doesn't it? I kind of like that feature. There's your taproot. So there you go. What's a carrot? Well, it's an example of a taproot. There you go. How many embryonic leaves would it have? Oh, probably 2. Whereas our fibrous root lacks that. Excellent. Whoop. Okay, quick little mention here about woody and herbaceous plants. Herbaceous means sort of succulent. They can lay down xylem, but it doesn't have to be heavily reinforced, right? It doesn't have to have a strong sort of cellulose outer neck on it. It doesn't have to be like that. So when we look at our woody plants, they do lay down that heavily reinforced xylem. Trees, shrubs, vines, etc. And some don't. Some lay down much more lightly reinforced tissue, and it doesn't go that crazy with the cellulose necking around the xylem, as it were. Well, if you've if you've done any gardening, okay, you'll know that plants have different lifetimes. I just go through this pretty quick. There are annuals, biennials, and perennials. Now, annuals means basically 1 year. So what we're looking at, and I got to get rid of brown because I'm just tired of the brown color. Let's go back here. Annuals will live like 1 year. Biennials think of them as using their first year to kind of get going and develop their reproductive structures. And in their second year, that's pretty well the end of their life cycle. And perennials can just keep on going, right? Trees, the tree of life. I should jump out. I wanted to show you that. Kind of spontaneous, but what the heck? The tree of life. Okay, tree of life is a 1,000-year-old tree. You get Yggdrasil from Greek mythology. Let's see if I can get this. There we go. That's the picture I was looking for. Check out this tree. A 2,000-year-old living tree. It's amazing. They've dated it. Some of these trees live for millennia. There's a sequoia. It's kind of redwood. You can tell by the cone-shaped um, stem. But these trees can be extremely long-lived. 500, 1,000 years old. And these trees, I mean, look at the elephant for perspective to see how big this one is. Okay, so thought it was worth showing you. So there's your annuals, right? You have to plant these pretty well every year. You go to the gardening center. You put these in your garden. It doesn't mean they can't drop seed. Things like poppies, they'll regrow every year, but it's like a new plant, right? Biennials. So, I like these. You've got your irises, things like that, and it takes them the first year to really develop their bodies, and then by the second year, that's when the flowers come out and they pretty well die. But don't be too sad about that because, hey, they drop seed and then you get new generations, new life cycles. Okay? Pansies tend to do this. And then there's your perennials, right? Your long lived plants. And this isn't just about angiosperms. This comes up with evergreens as well, conifers. Um, Plants can have different life cycles. That's how it goes winter, summer. Okay, that takes us to the introduction of 22.5. Now, what I'm going to do in the course is I'm going to go in a little bit further. I think it's worthwhile to let students see what are the tissues in plants. Like, get to a little bit of botany, get into the detail. Okay? Um, I will have, to that end, a lesson on soil and roots, stem and leaves, and a little bit of a discussion about photosynthesis. Because how can you really look at Kingdom Plantae and not talk or describe or get into detail about photosynthesis, right? Not college or university level photosynthesis. We're not going to look at the light and the dark reactions, and no, we're not going to go that far. It's great stuff, but not at the grade 11 level, folks. Okay, so that's it for this lesson, 22.5. Hope you guys enjoyed. Have a good one. Mr. B, out.